Okay, so I don't know how I clicked on that, and that's not where I want to be either. Okay, so discussion board for this module. I usually actually purposely do this one kind of late because I find it's good right before we get to disorders. So it's basically a what they call a web scavenger hunt. So you guys will find a website, can be a page, can be a video, can be an animation, can be a game, could be a quiz or flashcards. Students love that right before the exam, right? Flashcards are always great. Interactive type things, some related somehow to immunology. So one of the things I thought I had in here, but I'll add, as I know I wrote it for my other class, is that some of you guys may be interested in diseases, right? And we're going to be getting into diseases after this module. So like if there's a particular disease that interests you, especially an autoimmune disease, right, where the immune system is attacking self, you know, find some information about it or a video about it or something like that to share with us as you're learning probably what it is that causes that problem. I actually, what are the chances? Last semester I had 2 students that had the exact same autoimmune disease in 2 different classes. And both of them shared that with the class through this. And we all got to learn something new, which was really kind of cool. So one of them had lost their spleen to the disease and the other one had not as of yet. And it had to do with their platelets, their immune system attacking their own platelets. So it was really interesting for them to have shared that with the class. But if you know of somebody that's suffering from something and you want to know a little more about it, this is a good opportunity to do that little bit of extra research. And now that you guys have a little bit better understanding about the immune system. But any disease in general, right, there's an immune component too. So if you're really interested in diseases and you want to know, like, how is it that we get sick? How does the immune system fail? Or how does the immune system eventually win? Or both, right? These are things that you could investigate as part of this assignment. Does that make sense to you guys? So as always, right, you're going to share your stuff. And I actually shared and gave you guys an example of one. And I like this one because they draw similar to the bonus thing that I had assigned for you guys that I wanted, I think, for you guys to get done by today. So remember, under modules, it's actually in this module, the bonus, right? That is today. It says 0 points, right, because it's bonus. Otherwise, it would calculate into the points in your grades, right? This is extra points, right? So you can get up to 10 points. And remember, I added where you could take a picture of your notes and upload them. So one person already did that, and it worked fine. So that's an option for you too, instead of having to type notes to get your points. So the other thing that was going to require you to take notes was, unfortunately, the talk that was supposed to be yesterday that ended up getting canceled. I've been told it is going to get rescheduled, so hopefully as soon as I know that date, I'll pass it on to you guys, and we can hopefully still go to that. And then I have one other bonus that I'm noticing that I didn't add my Connect link to you guys. Not external. External tool. Where is it? There it is. So at this point, you should have definitely finished the innate Connect assignments, right? But I added something new, and I don't know if any of you guys have played around with this. So I'm going to go ahead and link out to it real quick. I added a Learn Smart, and everybody this semester, because everybody signed up for Connect, and the only option was with the ebook, because they're getting rid of the other option. And the reason for that is that Learn Smart is linked to the ebook or their smart book or whatever they want to call it. So you notice I've added a new assignment that's also bonus to help you get ready. Instead of doing didactic PowerPoints, I'm going to try something new, right, for next week's class. So you would click on the assignment, And I guess I needed to click student view beforehand. Student view. I always think it's on the next page, but it's on this page. Come on, student view. Alright, so what it looks like for you guys. So it actually loads the smart book. And so what you'll do is actually read and answer questions before class. So of course it's taking forever to load. So this assignment will take you right to Chapter 17 on disorders. At least it should. Yes. So what you do is you read the highlighted sections. So notice it's kind of grayed out. And I think it might be taking me to where I left off. Which is not good. But you'll see this practice button. So after you do a little bit of reading, you would practice what you've been reading. And so before you answer the question, it wants you to know, do you know it? You think so, you're unsure, or you have no idea. And this is going to give you guys an idea of like how effective your reading and your studying is. And unfortunately sometimes they have these fill in the blanks. So anaphylaxis is blank hypersensitivity mediated by repetitive exposures to an immune system provoking substance. And I have to say I'm not sure, right? But look, you can click on read about this. Ah, there, now it's going to the reading, right? So you would read, right, and the answer would be somewhere in here. And look, you can just click back. So you can read, right, try to answer the questions, and then just keep doing that until this assignment is complete. Okay? And I only made it 10 minutes. So, I mean, if you're a good reader and comprehender, it should only take you 10 minutes to do this assignment. So I want you guys to play around with this. Encourage you to do so. I'll give you 10 bonus points if you complete the assignment. Right? But you have to complete it before class next week. So, and other instructors say students rave about this. I've tried it. I've got some more tips. We'll try it again, right? Whatever might work for you guys, I'm willing to try it out. And apparently, forget where it is. Oh, here. So here's your options. Read. Which I don't know why. I guess it just leaves off where I was reading instead of the assignment. But there's this recharge where, like, after you do this assignment later on when you're getting ready to study for the test, it'll go through the stuff that was hard for you. Like the stuff that you definitely wrote, I don't know, or I'm unsure, or I don't think so. Right? So it's adaptive. It like adapts to you. Hence why, again, I think it keeps going to the reading where I left off instead of like where it would be for this assignment. Because it tailors to you. Does that make sense to you guys? Okay, all right. So give that a try for this next chapter. And you guys all have this, right? So you should all be able to do it. It's not questions, it's topics. So the thing is that it's going to re-ask you the question in a different way if you get it wrong. So you have to get through 10 topics in order to complete the assignment. Does that make sense? So the number of questions is going to depend on you. So it could be 10 questions if you get them all right off the bat. Or it could be 20. And then there's a little woman that'll speak to you too. Like if you keep not doing so well, she'll be like, read. Or after you've been reading so long, she'll be like, practice what you've read. She'll talk to you. You can turn her off though. If she annoys you. Okay, so I just wanted to point that out to you guys, and I'll put an announcement. I'll remind you guys. So as far as assignments go, you should have already completed the bonus. If you haven't, get on it. And then that bonus activity, right, watching the videos and submitting your notes, right? This is a new bonus, and then we have the new discussion board, which I turned on but forgot to put in our module. So there it is. Okay, so plenty of stuff to keep us busy, and obviously I moved the test back, right? So Thursday we don't have class because of spring break. I don't know about you, but I really need a break. Granted, I'll be working from home, but, you know, I won't have to come here. So, you know, it's somewhat of a break. I told my husband, I was like, oh, I have Thursday and Friday off. He goes, yeah, right. He'll still be working. I'm like, yeah, I will. But I won't have to go into work. You know, I can stay in my pajamas. It's all good. So we have that. So the test, so next week we'll finish up disorders. I already went ahead and posted that lecture for you guys. And then we'll have our test the following week, which means we'll really have to motor on the last bit of stuff. But I find students do better in it because it's diseases and, unfortunately, relatively a lot of memorization, but not necessarily that much understanding that has to happen. But also, most of you guys have a general understanding about disease, right? Most of us do. And definitely, hopefully now a more profound understanding now that we understand the immune system. At least I hope we do, right? Test is next. Oh, yeah. So there are other Connect assignments in Connect that aren't linked to the modules. There's one on antimicrobials, one on epidemiology, and usually I introduce those next unit or module. And there's stuff that you guys need to go over. I'm not going to spend time lecturing on, though, because I'm not going to test you directly on this material. But you guys do have to demonstrate that you've gone over the material. So what I do is I just give you Connect assignments related to that. So that's the other. That'll be introduced after this test. But that doesn't mean you can't start working on them now. But I don't foresee you guys doing that because you're going to be concentrating on this test. And I'm 100% okay with that. That's what I want you to do. Okay, so let's see, adaptive. So we were going over last time, and I wanted to do a quiz, but I also am concerned about getting through this lecture, so let's see. We might do a quick on-the-fly little poll everywhere quiz if we get through this. But you guys always have your Connect assignments, right, that help keep you on track. I'm reaching for a water bottle that does not exist this morning. That really sucks. Okay. So last time we were really talking about that cell-to-cell communication, and I find, again, that bonus assignment. Is it an extra? Yes, thank you. Thank you. So, we were talking about that really intimate cell-to-cell communication. So, different receptors that have different names do that. And so, I'm going to try to, like I said, kind of quiz you at the end, even if I don't do a poll everywhere, just a quick, reminder. So, where do these interactions happen, though? Where do these special cells shake hands, exchange information, right, and go on about their stuff? So, there's a whole system in our body of vessels that are to help this engagement and also to make sure that the bad guys don't get into the bloodstream. So we don't want that fluid that leaked out during inflammation into our tissues to just go back into the bloodstream, because it could contain viruses and bacteria, and we don't want them to get a free ride on the superhighway that is our circulatory system. So instead, they go into the lymphatic system. So this is our drainage system. It actually starts in your tissues. It doesn't circulate, it drains. And as you can imagine by these names, secondary lymphoid organs, primary lymphoid organs, we're really focusing on this interaction of lymphocytes, right? And what are our two main lymphocytes that are really important for our adaptive immune response? They're given initials. T cells and B cells, right? The T cells and B cells that we talked about last time, right? So this is where, it helps with the engagement of interaction for these. Oops. I don't know what I just did. I hit some wrong button, as usual. So as we said, the lymphoid section is designed to bring the B cells and the T cells in contact with the antigen in order for us to mount that response. So it includes the vessels, the secondary lymphoid organs, and the primary lymphoid organs. And what we could really also add to this list is lymphatic tissues. So the primary lymphoid organs, I actually went over this last time, but very quickly at the end. So this is where the white blood cells are born. So where are white blood cells born? In the bone marrow. And then there's one of our lymphocytes that must travel to another organ. And because this one really needs to be able to communicate cell to cell, between cells in our body, and really know the difference between healthy self versus non-self and unhealthy self, right? So these would be our what? These are T cells, and they're named after what organ? The thymus. Now this organ atrophies after puberty, which means it decreases in size. And so at one time scientists believed that it stopped functioning. In truth, it's still functioning. It's just not at the high level that it was functioning previous to puberty. Because when you're first born, you haven't seen anything yet, right? And so you need lots and lots and lots of T cells for your adaptive immune response. As we age, right, we've been exposed to a lot of things. We've got a lot of memory T cells. We don't need to keep producing a lot of naive T cells that haven't seen antigen yet. So the workload decreases, so it makes sense that the organ itself decreases in size. But it's still functioning. Just not at the high level that it was previously. Now, all the green vessels, right, as I said, they start in the tissues, and they're going to drain the fluid from the tissues. And they're going to keep combining. As they go, they're going to be filtered by lymph nodes. These are secondary lymphoid organs. These are what I call the homes of a lot of our B cells and T cells and macrophages, and where dendritic cells are going to travel to. So as that fluid is going through, it's filtered by the macrophages that are there. And if it picks up anything, it's going to present that information to the T cells. And the B cells may even interact with the antigen. There's one organ, secondary lymphoid organ, that's chock full again of lymphocytes and macrophages, is your spleen. Your spleen's job is to filter another fluid of our body. What does it filter? It filters your blood. So it's the one organ you can actually live without, and I don't think I put the answer here. No, I didn't. But then you're at risk for bloodborne pathogens. You're at greater risk. Some of the other functions of recycling components of damaged red blood cells gets taken over by your liver. And actually, as a baby, it's pretty much your liver that takes care of that job. Anyways. So the lymph nodes, right, these are organized to the organelle level, right? You have tons of lymph nodes. You have more of them around your gut than any place else because, again, this is a large surface area in which invading microbes could get in. So we definitely want to be able to protect against that. You have large concentrations in all your axillary points, so in your armpit region, in your groin region. But you have them scattered throughout. You have much more concentrations in those particular areas because all these vessels go into two ducts, your left and right lymphatic ducts. These two ducts dump the fluid back into your subclavian veins, so back into circulation. So we want to make sure that it's been filtered and cleaned extensively before it gets there. But we do need to put that fluid back into circulation. If you've ever had swollen glands in your neck, right, those are your lymph nodes, usually. Right? And again, that's a sign that your immune system is responding to infection. The numbers of cells in those glands has increased in response to the infection that you're dealing with. I unfortunately got an infection the first time I got to Disney World. It was horrible. I had to go one day to some, clinic to get antibiotics. In the pictures you can see my whole face and my neck is all swollen. It was horrible. I was like, of all the times to get sick like this, right? Walt Disney World. And when you palpate, when you touch those, they're usually sore, right? And that's actually a good sign, right? It means, again, that your immune system is responding, right, to some invasion that's happened. When lymph nodes are hard and they're not tender, unfortunately, sometimes that's a sign of cancer, right? So you want that when the doctor palpates that it's tender, right? You want to feel like you want to hit them back, right? You don't want it to be such that it's not tender. So in addition, because again it's a major entry point, right? Your mouth and your nose, you have tonsils. You have just one tonsil, although it's commonly referred to as your adenoids, which is plural, but it's actually only one tonsil. In the back of your nose, and remember your throat extends up it connects your nose to the back of your mouth and then goes down into your esophagus. It's referred to as your nasopharynx. So in the back there is your adenoid or your nasopharyngeal tonsil. And unfortunately, it's also right between your statiary tubes. What are your statiary tubes, Drain? Your ears, your inner ear. So for some people, if that swells up, it blocks the statiary tubes and impedes the flow of fluid out of the inner ear. And a lot of times how our inner ear gets infected, again, is the fact that it's basically linked to our nose, right? Those statiary tubes dump into the back of our nose, into the nasopharynx region. And so things travel from the nose up the statiary tubes, and that's how they usually infect the inner ear. Anything that stops that drainage of stuff out of there can, really cause serious infections. So for my son who has bad allergies, before we realized that too, little kids, their statiary tubes tend to be more horizontal, and as your head grows and you age, they pitch. So drainage is much more easy. All right, so little babies get ear infections, unfortunately, partly because of anatomy and partly because of the environment. Or if you have other factors like my poor son with allergy, then you're always having so much mucus, so much stuff, and it's getting trapped in there. So he had tubes put in his ears when he was 2, and they also removed about 80% of his adenoid because it was swelling up and blocking his statiary tubes, right? So sometimes, I wish I was like, I kind of wish I had that surgery done, right? Probably would help with some of my nasal issues that I deal with. But as an adult, I don't at all want to go through that if I don't absolutely have to, right? So and then you have your palatine tonsils, which are actually in your throat. And sometimes those will swell up for people, and if they continually swell up on a pretty regular basis, they may remove them. Because again, it will block your throat, right? You kind of need for stuff to get down the throat. But again, they're there, to help combat stuff that gets in right on site. You have one associated, a lingual tonsil, and I have to say I've never heard of that one swelling or being removed. I think it's pretty much not as active as your other tonsils, your adenoid or your palatine tonsils. And then your appendix, right, which is associated with your intestines, has a very large concentration of white blood cells, B cells, T cells, and macrophages. The problem is that sometimes stuff can get stuck in there, right, and it can get really inflamed very easily because of the large number of white blood cells there. The bad news with that, if it bursts, you end up with a hole in your intestines, right? And now you have stuff that's supposed to stay in the intestinal tract getting into your abdominal cavity, and can be very life-threatening type condition. So in addition to the organs, right, where we're organized to an organ level, you have tissues that are associated, again, with major entry points. So you have mucus-associated lymphoid tissue. So this is, of course, associated with your mucus layer. So lining your entire intestines. And those of you guys that are in anatomy and physiology, right, you look at a cross-section in A&P2 of the small intestine under the microscope, and you see these congregations of white blood cells, what are referred to as Pryor's patches. And you have specialized M cells, right, that will purposely sample antigen from your intestines and present it to macrophages. The dendritic cells can actually reach into the intestines and grab stuff. And they're going to present this information to the T cells and the B cells and mount a response. Now remember, B cells are going to make antibodies, right? When we're associated with the mucus here, like the Pryor patches, what antibody class are they mostly going to make if we're going to secrete it into our mucus? We're going to make IgA, right? Because, again, the environment of where they're at and what is probably needed, what level, what type of protection, right? And we know for our secretions, that's IgA. Now for our skin, again, that's a major entry point. So scattered throughout your skin, you have patches of T cells and B cells and macrophages and dendritic cells that are there to defend right on the front line. Again, not organized to an organ level, but it definitely is a tissue, a lymphatic tissue. And so the abbreviation we use for that one is SALT, which I find kind of funny, right, because our skin is salty, right? But in this case, we're talking about skin-associated lymphoid tissue. So again, these lymphatic vessels start in the tissue, and they're gonna grab that excess fluid that goes into the tissue during inflammation. We don't want it to go directly back into circulation because there could be pathogens. And so we're going to bring it to where we can deal with it, right? So we're going to filter it all along the way through lymphatic vessels. Through lymph nodes. So what happens, right? We talked about this special handshake that happens, right, where a B cell recognizes a specific pathogen, right, by its unique epitope, by the B cell receptor, which is basically an antibody. And which antibody class is on the surface of the B cell? What's the very first antibody class you make? IgM. IgM. So once that happens, what are the sequence of events that have to happen? So note that this is for both B cells and T cells, hence why I have T cells in bold. So although we're going to watch this animation in a second that concentrates on what happens for B cells, because, it's pretty cool, and antibodies are awesome. This also happens for T cells, right? The same process. But of course, what they do is a little bit different from what B cells do, right? B cells are going to generate antibodies. T cells are going to attack other cells if they're effector, cytotoxic T cells, or they're going to help out the B cells, or they're going to help out the macrophages if they're helper T cells. I always have this on mute and forget. Oh, no, somebody has this on mute. That's really not going to do us any good. Sorry, I went all the way up.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
So what they just showed you there, the first time, right, there weren't any T cells needed. And in that case, we're just going to produce the normal first antibody class that we normally produce, which is what? IgM. And what's the limitations with IgM? What does it look like? What does IgM look like when it's secreted? Does it look like what they're showing here? Right, it's that big, huge pentamer, right? That mega big antibody. And what are its limitations because it's so big? It pretty much stays in the bloodstream, right? It can't get out. It's too big, right? This one doesn't cross the placenta, anything like that, right? So it has limitations. But, you know, it's good at activating complement, right? It's great at cross-linking, right? Agglutinating, clumping stuff up. It's going to stop if it binds to something. It could neutralize a toxin or a virus from getting into our cells. Right? So it's beneficial in that way. But sometimes we need the other classes, right? We need greater protection. We need IgG that can get out of the bloodstream and cross the placenta. Or maybe we need mucosal protection, right? Maybe we need IgA, depending on what we're dealing with. In order for that switch to happen, you need that interaction that they just showed of a T cell interacting with the B cell. An activated T cell at that, where it recognizes that antigen, right? You need this interaction. Because in that interaction, again, notice they talked to each other, right? Interleukins, right? Those cytokines, those chemical messages. So they're relaying important information, like maybe doing what we refer to as class switching, where we're not going to make IgM. We're going to now switch and make IgG, right? Or we're going to make IgA, depending on where this interaction is happening and what is needed and what type of invader they're dealing with. So they're pretty intelligent little communications that happen there. And we refer to that as T-dependent, right? You are depending on the T cell to help you out in this case. Right? So some antigens, they can be T-independent, which means that the B cell is going to go ahead and produce IgM. Where others, right, we're going to produce IgM, but we're also going to get that T involvement So it's going to be what we refer to as a T-dependent antigen. To get those switching, to get that different level of interaction, you really need that T-cell involvement. And it really depends on the antigen, right? What are we up against, right? Is IgM going to take care of it, or do we need a little bit more sophisticated assault on the bad guy? So clonal selection. So why clonal? Why are we saying clone? What does clone mean? It's an exact copy, right? So that B cell, remember, in the very beginning, they selected one of them, right? So here we have all these possibilities, right? And they're showing each one of these B cells with a different colored antibody on its surface to denote that each one of these B cells is expressing a different antibody. So it's only the one that interacts with that specific antigen during this challenge that we're dealing with that's going to be activated. And once it becomes activated, it proliferates, right? All cells, all lymphatic cells, once they become activated, they proliferate. They clonally expand, right? So we don't just have one B cell that recognizes, we have thousands of them that recognize that specific antigen, right down to its epitope. So could, for an antigen, since antigens are made up of several different epitopes, could we activate another one of these B cells potentially? Yeah, we could probably activate all these guys seen here potentially, right? But each one of them is interacting with a different epitope on that antigen. Because remember, antigens are quite complex, right? Even a protein like insulin or a protein like penicillin. There are multiple sites where an antibody could bind. So we're going to produce lots of different antibodies. But that one B cell is only producing one specific antibody. It's going to expand, and in order to produce antibodies, what cell type did we say it turns into? What is a B cell that's secreting antibodies called? Plasma cell. And remember, antibodies are what type of molecules? Are they sugars? Are they proteins? Are they nucleic acids? They're proteins. They're proteins. And so therefore they're made by what? By ribosomes. And in this case, ribosomes associated with the endoplasmic reticulum. So the rough endoplasmic reticulum gets huge in these cells, y'all. Their cytoplasm gets enormous because they're producing a lot of secreted protein. And then you're going to have some, as we said, that are going to stick around and be our memory cells. So each time, so this is just schematically, right, all together in one diagram, right? One of them is selected that recognizes the antigen, right? They're going to proliferate, and then they're going to differentiate. So we're going to get some plasma cells that are actually going to secrete antibodies, and then we're also going to get memory cells. I want to go back to this other one. So B cells or T cells, right, are able to recognize a functionally limitless variety of antigens. That's because we produce millions of different B cells and T cells, all of them with different receptors, right? Each B cell is unique. It has its own special antibody it made. Each T cell is unique. It has its own special T cell receptor it's made. Each of those only recognizes and responds to a single epitope. So as we saw, only one of the B cells actually recognized that antigen at that site. And this is where the time delay is a problem for us, right? Is that we've got to either generate or find the B cell and T cells that recognize the specific antigen we've been exposed to. And this can take up to 2 weeks. If we succeed even. There are times that we don't even succeed in this task. This diversity recognition is random, right? So we don't get an antigen, and we don't have B cells going, ooh, let me see how I can make my antibody fit this. It doesn't work that way. The antibodies randomly make the B cells randomly make those antibodies. The T cells randomly make a different T cell receptor. So we're basically we're basically hoping we win the lottery each time this happens, right? We're looking for that we've picked randomly the correct molecules to come together to form those receptors that will recognize a particular antigen out there. So why? It's like, wow, that's pretty scary, right? Because this is our life. This isn't money, right? This is our lives that we're betting on right here. Why do we have this system where we're basically leaving it up to chance, right? Why don't we come pre-programmed that everybody knows how to make an antibody against E. coli 0157? Why don't we have that level of specificity? Well, think back to our genetics chapter. Did E. coli 0157 always exist? No. He existed because he acquired a particular toxin from Shigella, right? He genetically changed by gene transfer in that case. But do they mutate? Do they change? Yeah. So if we didn't have the ability to change, we would be eliminated, right? Because we wouldn't be able to keep up with the newest, greatest superbug out there. So this random process allows us to potentially recognize a limitless number of pathogens that exist or may exist in the future. So as you saw, there are interactions that happen, right? There's communication that happens between the cells. It isn't just the receptors. There's communication that happens. And the good news for you guys, again, not a graduate level immunology class, so we're going to keep it basic, right? We're not going to go into all the specifics. But know that there's a lot of interaction that's happening there, more so than the detail we're discussing in this course. So once that interaction happens, that conformation happens, they're gonna multiply, right? Whether it's B cells or T cells, they're gonna multiply, right? And make copies of themselves. So now instead of just one guy that recognizes the bad guys, we've got thousands of them. Some are gonna leave the secondary lymphoid organs and migrate into the tissues. And they'll continue doing what they need to do until antigen is gone. And then once the antigen's gone, most of those workers, right, those plasma cells, are going to undergo apoptosis, programmed cell death, right? Are we going to get inflammation when they die? No, because remember, apoptosis is programmed cell death without inflammation. So their job is over. We're going to kill them off. Why would we kill off an awesome plasma cell? I mean, they produce antibodies. Why are we killing them off? We're not going to keep them? Why don't we need to keep them, for one? Because we have memory cells, right, that can be activated to turn into plasma cells in a moment's notice. Yeah, you don't want to be producing antibodies when you don't have to be, right? If you've taken care of the problem, then we don't need to keep making antibodies. Not to mention, antibodies are proteins. That's not cheap to make, right? You've got to eat a lot of food, generate a lot of energy to keep those guys happy, right? You don't want to support what is not necessary, right? That would be a waste of energy, plain and simple, to maintain and continue those plasma cells when it's not needed anymore. All right, so that's that. So naive cells, right, when we talk about lymphocytes, they have an antigen receptor, but they haven't encountered antigen yet. They're naive. They don't know that bad guys exist yet. They haven't found their bad guy. Once they become activated, right, they've interacted with the bad guy, they're able to proliferate, they have bound antigen, right? They've interacted with the bad guy. Effector cells are descendants of those activated naive cells. And they're ones that are going to do something. So B cells turn into plasma cells which secrete antibodies. T cells become activated helper cells that go and help macrophages and help B cells. Cytotoxic T cells go out and kill other cells that are expressing that antigen that they recognize. And then for all lymphocytes, whenever we get it activated, right, we produce these effector cells, we differentiate into these effector cells. We're always going to produce memory cells, right? Not all of them are going to become effectors. Some of them are going to become memory cells. So they're long-lived descendants of those activated lymphocytes. They've seen the bad guy. And this is what really helps us with our second response. So if we see it again, we're going to respond that much faster. We don't have to find a naive cell. We've got memory cells. They remember this bad guy. So we really take advantage of this ability of memory cells, of remembering what we've seen, when we vaccinate. When we vaccinate, we get the body to respond. We get it to produce antibodies. We get it to produce memory cells. So that way, if you see that antigen again, your response is much faster. You're not waiting 7 to 14 days to find a naive T cell and naive B cells that recognize that antigen. You already have memory cells. And each time this activation happens, you produce more memory cells. Right? This same progression happens every time. So what's the role of the TH cells? In this case, we're referring to helper cells, right? In B cell activation. What's the difference between primary versus secondary response? What are the similarities? We've already mentioned T-dependent versus T-independent, right? This is where you either need or don't need the T cells to help with the B cell activation. So again, B cells can directly interact with an antigen by their B cell receptor, which is IgM antibodies. They're going to take it inside, they're going to internalize it, and they're going to present it. Remember on MHC class 2, only antigen presenting cells can do this. A B cell can do antigen presentation. Who else can do antigen presentation? Who's going to physically take in an antigen, process it, and present it to other cells? Macrophage, and one more. Dendritic cells. And those guys are very important for T cell activation. But here we're looking at B cell activation. So a B cell has got to present that antigen that it took in, that it recognized. And it needs that confirmation sometimes from the T cell, right? This would be for T-dependent antigens. So remember, the T cells have their T cell receptor that are going to help recognize that specific antigen, that specific epitope, that piece of the antigen that the B cell is presenting. Now, helper T cells can communicate with MHC class 2 because what CD marker do they have on their surface? What makes them a helper T cell? CD4, right? And they actually show it here, right? That little molecule next to their T cell receptor is CD4. So again, notice he's recognizing this antigen the B cell is presenting. So he's sending signals, cytokines, right? He's communicating with this B cell. This is going to activate this B cell. It's going to proliferate. And it's also going to help that cell determine, too, what class of antibody it's going to make. Because now it could switch. It doesn't have to stay stuck with IgM. It could go to one of the other classes. Here, notice no interaction is happening, right? So we stop here. Right? They're not recognizing each other. So no proliferation is going to happen of that B cell. So as you can imagine, as I said, this takes time, especially the first time you're exposed. So the very first time you ever see a particular antigen, there's going to be a lag period. Now here it says 10 to 12 days. The average that I see written more often is 7 to 14 days. But still, that's significant time, right? That we're not producing any antibodies because we're trying to find the B cell that recognizes, and we may even need a helper T cell to help us out. Right? So in the beginning, we're always going to, as I said, produce IgM. We'll switch to maybe something like IgG if you get T cells involved, right? And they're like, yeah, we really need, G. Or maybe we need IgA. And those numbers are going to increase, and then they're going to drop off, right, as the antigen has been eliminated. Then if we see the antigen again, right, notice that IgM, but boy, because we made that switch last time to G, we took off like gangbusters. I mean, look at that. It's an insane number of IgG. Really fast, too. And we had no lag time here, right? Boom, antigen. Boom, antibodies. Why no lag? Because the memory cells were activated this time. Here, it was naive cells, right? Naive B cells and T cells had to be activated, had to be found and activated. But that same antigen, if we see it again, we're not trying to find and activate naive T cells and B cells. We already have memory T and B cells. So our response is fast and very aggressive, as you can see, right? We're not messing around the second time. So what's similar? What's similar between primary response and secondary response? What has to happen for us to produce antibodies? We got to activate T cells and B cells, right? That's going to happen both times, right? But the first time it's naive B cells and T cells, right? That have never seen antigen before. The second time, who's getting activated? Memory cells. That's the significant difference between primary and secondary response. And why vaccination really helps us out, because vaccination is probably the first exposure, right? We'll activate those naive cells so that we produce what that's going to help us out with the secondary time you come exposed. Memory cells, right? Those very important, awesome memory cells. So B cells, as we said, they differentiate to form plasma cells. You can definitely see, as I said in this picture, their cytoplasm gets enormous, right, because their rough endoplasmic reticulum increases to actually produce all those antibodies, which are proteins for the most part. So that concentration of antibodies as it goes up, right, a concentration is referred to as a titer. So those of you guys wanting to go to nursing school and such as that, if you don't have your vaccination records, another way you could check to see if you've been vaccinated or exposed to the different things that they want you to be protected against is they can run a titer, right? So they can run a titer on your blood and determine about how many antibodies you have against the different pathogens they're concerned about, right? So for measles and mumps and rubella. And they usually do this when you're pregnant too. They want to know where your titer is to know what level of protection you're going to have to be able to protect your fetus. If your titers are really low, they're going to suggest after pregnancy that you get boosted for particular vaccines such as that, so that you're protected for your next pregnancy if you decide to do that again. For some of us, once was enough. Yep, once was enough. Not to say that being pregnant was bad. Being pregnant was actually great for me, but taking care of them afterwards is a lot of effort.
So like with syphilis and HIV, whenever it gets to a certain low state, they call it undetected for both of them, but they still say that you're, so it's like with HIV, you're still positive HIV. With syphilis, you can't spread it, but it's considered deactivated. It's like, they say it's like chicken pox, I guess.
Right. Yeah, I don't know this.
Difference? Like, what is that? Like, how do they? I don't know how that works. Like, how do they read it? Like, as like.
Right.
I don't know. Like.
So the different tests that we do for the different diseases, it depends too on what you're testing for, and how you're testing for it. So different tests have different levels of accuracy, right? And that's the kind of scary thing with HIV, is that you could come out negative but actually be positive because your numbers are so low, they're not detectable by the test. And actually, in emergency rooms, I can tell you, at one time they would do, I think, the antibody test, which obviously you'll have an active infection if you have active antibodies, right? And they could, there's another test where they actually can detect for the virus. And again, depending on, where you are in the state, you might not have that much of a viral load in whatever sample they're taking. I think they might do a blood sample in that case in the ER. But they are going to test both nowadays. Like, they test for both. But again, they're using a test that the sensitivity level isn't necessarily quite there. But then they're going to run additional tests, because they don't want to tell somebody that they actually have something unless they're absolutely sure. So there's different types of tests that are going to give you different levels of sensitivity and accuracy.
The rapid tests, like oral swabs and stuff, they test for the antibodies, and they say, like, if you've already been infected with, it's similar for syphilis and HIV. If you've been infected with syphilis and you had that treatment, then they have to test you through the blood and have to wait because they said that the rapid will pick it up like that. And they'll think that you're, like, have a high titer count.
Right.
Then they said same with HIV. If they test for the antibodies and it comes up positive, they have to do a blood test for the antigens.
Right, exactly.
It's kind of interesting to me. So.
Yeah. So because antibodies are going to stay much longer after infection, right, than the actual antigen itself. Right. So, so the other thing too is we talk about these B cells clonally expanding, right? So they're going to make copies. But when they do this, they're actually going to tweak their antibody a little bit, right? They're going to make minor changes. They're actually going to cause mutation on purpose. And the reason for that is they want the best possible fit. But again, this is random, right? So they randomly may produce an antibody that has a much better affinity, much better binding to the antigen, right? And so they're able to recognize this. So just for understanding purposes, right, they code it here with 2 pluses. Where this guy is binding but not as strong. So he's not going to get the signal to continue, right, because we want the best possible binding. So if we had to pick between these two guys, this guy has the best interaction with the antigen, the best affinity, the best binding. So he's going to go through a round of replications. In that round, as I said, they're going to mutate their DNA in hopes that they're going to get a stronger bonding. And so in this case, again, a much stronger bonding existed, so that lineage continues So they continue to tweak this even as we're proliferating. But again, it's random. So sometimes we're going to get better, sometimes we're going to get worse. But obviously we're going to put our energy towards the ones that are having the best affinity for that antigen, right? And not continue those lineages that aren't as effective. And so that's referred to as affinity maturation. That affinity is how well it binds to the antigen. So again, these are proteins, right? So where's that information encoded on how to make antibodies? Where in your cell is all your information stored? In the nucleus. In what molecule in the nucleus? In the DNA. So in the DNA, right, is the information on how to make an antibody and the information on how to make all 5 classes. So when we switch, so say in the lymph nodes, those B cells get a signal from the T cell that says switch to IgG, right? IgM's great, but we really need some IgGs. They're literally going to change their DNA. They're going to cut out the section for IgM and go so that they're going right to IgG. And again, if maybe they're mucus-associated lymphoid tissue, their B cells there, the most common switch we're going to make is to IgA, right? So that we can secrete that into the mucus. So they would literally cut out M and G and E and make A instead. In other words, basically once that B cell has made that switch, can't go back. Stuck. So secondary responses, we said, it's those memory cells that are really responsible for that very swift, no lag time reaction. And vaccination helps us, put us to that second spot instead of being at primary response for certain antigens. So how is it that a B cell or a T cell, but we're going to again look at B cells, how is it that each B cell is different? If the information on how to make an antibody is encoded in our DNA, shouldn't they all make it exactly the same? You would think, right? Certain parts we're going to make the same, right? Those constant regions so that the light and the heavy chains can bind, and we're always going to make the IgA portion of IgA the same so the body knows it's IgA. But how do we make the variable section variable? How do we make each one of those different? We play the lottery with our DNA. So we've got information, but we're not going to use it all. We're going to randomly select sections of that information and put it together differently for each B cell. Each B cell plays their own lottery. They're going to pick pieces, and they're literally going to pick, right? Once you fill out that form for the lottery and you hand it in, can you go, oh no, I want to change my answers or my picks? No. Once you hand it in, that's it, right? So same thing for a B cell. Once he picks which part of the DNA he wants, that's it. Because he literally will cut out the other stuff, and it's gone. Yeah. So how do we do this? We do gene rearrangement. We literally rearrange the DNA, right? We pick different portions of it to create a different protein for each B cell, for each T cell receptor. Do we put them together precisely, perfectly? No, right? So if you think about a gene, right? You think about A's, G's, T's, and C's. One gene may be like 50 base pairs, right? A's, G's, T's, and C's. But we're not going to specifically even grab that whole segment. We're going to take maybe numbers 2 nucleotide down to, I don't know, 47 instead of 1 to 50. Right. So even the discrete segments aren't cut discretely. Does that make sense to you guys? I wish I could find a video out there that would show this. I have not yet. Been trying to come up with an activity too that works. So that's what we mean by imprecise joining, right? So we're not, even if they picked segment 5 and segment 8, they're not going to take the exact same section from 5 and 8 and put them together the exact same way. Now remember, for an antibody, how many variable regions do we have? How many proteins make up an antibody? One of them's really long. We call that what? The heavy chain, and one of them's shorter, so we call that one the light chain because it ends up being lighter. If you remember the antibody structure, both the heavy chain and the light chain have a variable region. So the combination of those two that form the antigen binding site is the combinational associations, right? So we're picking different numbers, we're picking different sections within those numbers, and then we have the combination of a light chain and a heavy chain. So this animation that we're going to watch covers gene rearrangement really well and combinational. It doesn't show the imprecise joining, unfortunately. But this is how we generate diversity, right? And so this is our last animation. And I think our last thing.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chains of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
But remember, each B cell only makes that one antibody it has picked, right? The next B cell it's generated, he picks his numbers, it's going to be different, right? Each time, even if they pick the same numbers because of the imprecise joining, they're going to be different. All right, so don't forget, those of you guys that may have come in late, I was trying to see about this. Yeah, most of this other, we still got a little bit more in this one that we got to go over. Natural killer cells. So we got a little bit more of this one, but we will start disorders next time. Make sure you do that bonus Learn Smart. Basically, make sure you watch the beginning of today's recording. See you guys after break next Tuesday. I have to hit done.
I was in the hospital all day. My sister